Dear students, in the last lecture we discussed about deletion type of mutation under the topic chromosomal mutations. Today we will discuss about the second type of mutation, duplication, as follows. Duplication is a structural change resulting in doubling of a genes in a section of chromosome of prokaryotes and eukaryotes. It is the occurrence of a segment of chromosome in two or more copies per genome. For the first time, the term duplication was coined by Bridges 1919, who discovered it in Drosophila melanogaster. The duplication is categorized into two, intrachromosomal and interchromosomal duplication. Interchromosomal duplication, here the duplicated segment of chromosome is incorporated in a non-homologous chromosome, and its example is transposed duplication. The intrachromosomal duplication is if a segment of one chromosome is incorporated in another homologous chromosome, either on the same arm or the different arm once removed from the original part of the chromosome. These are of four types: direct tandem duplication, reverse tandem duplication, displaced tandem duplication, and displaced reverse duplication. Now the first category is direct tandem duplication. Here the duplicated gene sequence lies just next to the normal corresponding section of the chromosome. Here I have drawn a chromosome having genes A, B, C, D, E, F, G, H arranged in a linear manner on a chromosome. Due to the duplication, the EF get duplicated. As you can observe here, this EF is repeated again as EF, and the sequence is as same as it was earlier in the arm. The direction is the same, and this type of duplication is called direct tandem duplication. So we can observe that the sequence of gene is as same as it was earlier on the chromosome. The second type is reverse tandem duplication. Reverse means in another or opposite direction. Here the duplicated gene sequence gets reversed and then lies adjacent to the normal gene sequence. Here, for an example, this chromosome having A, B, C, D, E, F, G, H genes. Now this E, F sequence of the genes is repeated or doubled, but the sequence has become now FE. As you can observe, the sequence prior is E, F, G. But due to the reverse type of tandem duplication, this is got inverted and now the sequence has become FE instead of EF. In another arm, this can be shown as CD inverted as CD, or DC can be read. Now the third type is displaced direct duplication. Here the section of gene sequence is not adjacent or continuous with the normal section. It can be on the same arm called intra-arm or in two different arms called inter-arms. Here the chromosome A, B, C, D, E, F, G, H is drawn. Now this EF sequence is doubled, but this is not placed adjacent to EF. Rather, it is being displaced to somewhere else, either on the same arm like EF. Now this EF is doubled after the GH. Rather than EF, EF, it is EF, GH, EF. Since it is on the same arm, this is called intra-arm, and if this EF is displaced to the another arm, this will be called inter-arm displaced direct duplication. The fourth category is displaced reversed duplication. Here it is as same as the displaced direct duplication, but the sequence of genes is reversed. Means the gene sequence in the interarm or intra-arm is reversed or gets inverted. Example, in this chromosome A, B, C, D, E, F, G, H, this EF is duplicated Since this is a displaced duplication, hence it will not be adjacent to EF. This EF will be duplicated somewhere else in the same arm or in the different arm, but the sequence of gene will get reversed, means EF will get reversed as FE, and this is now displaced to another place in the direction FE, and here on the other arm as FE. The fifth category is transposed duplication. It is an example of interchromosomal duplication. Here the duplication of the chromosome is found between the non-homologous chromosomes. Here the duplicated portion of chromosome becomes attached to a non-homologous chromosome. It can be either terminal transposition or interstitial transposition. Here we have two non-homologous chromosomes. A, B, C, D, E, F, G are the genes on first, and L, M, N, O, P, Q, R are the genes on second homologous pair of chromosomes. These two are non-homologous pair of chromosomes. During transposed duplication, this part of gene, either from the terminal or from the interstitial part, is deleted from one chromosome and gets attached to the other non-homologous chromosome. Here it is lying as BC between the LM and NO, and here in the terminal part. So it can be interstitial or terminal. Now the gene sequence has been duplicated. This arm has become longer than the original arm, as you can observe here. While this homologous pair, another pair of chromosome, this will be called a deleted chromosome since it has lost its two genes. So in a transposed duplication, we have one chromosome having deleted genes and other having duplicated genes. Now there are various genetical and cytological effects of duplication. First of all, we will discuss the genetical effect of duplication. Due to duplication, unequal crossing over is seen to happen. Secondly, the bar eye phenotype in Drosophila is due to the duplication of the genes. It appears in the chromosome 16A locus on X chromosome. Another example is polytene chromosome, which is found in salivary gland of Drosophila, where the chromosomes get duplicated many times and becomes giant. A number of duplication genes are found in peas and corns in case of plants, and this leads to the change in genic balance. This leads to abnormality in the phenotypic characters or diversifications in the duplicated members. It also affects the lethality of individuals. It increases or decreases the viability of these carriers. Under microscope, during the cytological study, we can found a loop formation during the synapse of the chromosomes. This loop formation is due to the duplicated part of the chromosome. During the synapse, the extra genes segment of the chromosome can be observed as a loop during the meiosis. Secondly, the crossing over in reverse tandem duplication results into a dicentric chromosome, means a chromosome having two centromeres. Example in maize. Since the two centromeres move to the opposite poles during the cell division, a bridge is formed which breaks at any point, and these broken ends are sticky and they replicate, and the broken pieces replication results in the formation of two sister chromatids which join at the sticky ends. This leads to the formation of isochromosomes. An isochromosome is an abnormal chromosome that has lost one of its arm and replaced it with an exact copy of the other arm. Now there are certain uses of duplication. Number one is We can study the chromosomal behaviors during meiosis. Secondly, it helps in the study of crossing over and its consequences. Next is that it is helpful in plant breeding. It helps to obtain high yielding varieties with high adaptability and disease resistance. This phenomena of duplication is more advantageous over polyploidy. New genes can be produced through duplication, which actually lead to evolution. Hence, it is very important for evolution and inducing variations in the organisms. In the next lecture, we will study about inversion and translocation in the chromosomal mutation. Thanks.